Huntington's disease is a rare monogenic terminal neurodegenerative disorder with a prevalence of 12 in 100,000 people of European descent. It is characterized by cognitive and behavioral changes in early stages, followed by involuntary movements and progression towards severe behavioral changes, dementia, and a complete loss of motor function. The sole cause of Huntington's is the dominant inborn expansion of glutamine-coding CAG repeats in the Huntington gene. This gene plays an important role in neural tube formation, axonal transport, and synapse maintenance. The striatum, followed by the cerebral cortex, are primarily affected. There is a large variability observed in the age of onset and the progression of Huntington's, which has been attributed to CAG repeat length along with the role of genetic and environmental factors. Studies have indicated that the length of the CAG repeat strongly determines age of onset, accounting for 50 to 70% of variation. The remaining variation is estimated to be 40% and is also thought to be at least partially heritable. The disease progression is not predominantly determined by the number of CAG repeats. Studies have also indicated that altered epigenetic and transcriptional regulation in Huntington's are associated with differences in individual presentations of the disease, along with environmental factors. Alterations on the epigenetic and transcriptional level can lead to changes in gene expression. Regulation can be both trans and cis-acting. Trans-acting regulation has a uniform effect on both alleles of the same gene, whereas cis-acting regulation only affects one of the two alleles. With RNA sequencing data, gene expression levels can be measured and compared between groups. In addition, RNA-seq data can be used to measure allele-specific expression and detect allelic imbalance, where one of the two alleles is significantly more abundant. This serves as a proxy for cis-acting regulatory events. Both gene expression and allele-specific expression can be used to find genes, pathways, and processes that are dysregulated within the Huntington's population. To gain insights in the regulatory variation within 20 Huntington's patients, we performed k-means clustering and network-based stratification on allele-specific expression profiles. Furthermore, we performed differential allele-specific expression and differential gene expression analyses between the detected clusters to identify genes, pathways, and processes that vary most in patients with Huntington's, thereby providing perspective on the regulatory variation underlying individual Huntington's manifestation and pathogenesis. There was no optimal number of clusters determined using the k-means algorithm on the raw allele-specific expression data. However, we found two clear clusters after performing network-based stratification of genes with at least one significantly imbalanced locus. Network-based stratification takes into account the downstream effects of gene and protein disruption by integrating protein-protein interactions. This way, allelic imbalance or cis-acting dysregulation in different genes belonging to the same neighborhood will be co-analyzed. The genes with differential imbalance and expression between the two clusters were further studied using GO analysis. Vesicle organization, vesicle-mediated transport in synapses, organelle fusion, and purine-related metabolic processes were enriched for the imbalanced genes. Mitochondrial, metabolic, and glial cell differentiation processes were enriched for the differentially expressed genes. We found 71 genes that were both differentially imbalanced as well as differentially expressed between the two clusters. Gene ontology enrichment analysis indicated a potential dysregulation of receptor-mediated endocytosis. A string database protein-protein interaction network was constructed using the 71 overlapping genes and known Huntington's-associated genes from DisGenet. This revealed two connections between our detected genes and the known Huntington's-associated genes HTT and GAP43. Specifically, a connection between CAGNA1B, a voltage-gated calcium channel subunit, and HTT, and one between MBP, or myelin basic protein, and GAP43. Reduced expression of MBP has been linked to early onset of Huntington's symptoms in animal models. The two identified clusters also show different expression levels of MBP. Cluster 2 showing a higher expression with a log2 fold change of 1.71. Concluding, our study has identified distinct patient clusters characterized by specific patterns of allelic imbalance as a proxy for cis-regulatory variation. These clusters display differences in allele-specific expression and gene expression related to several key processes. Importantly, our use of allele-specific expression analysis offers an additional dimension to traditional gene expression analysis, providing insights and promising leads for further in-depth exploration.